So I'm most grateful for this kind and generous introduction, for the welcome, for the invitation to be here, to all of you for coming to participate in this event. I was reminded by the first of the remarks made by the prorector of the fact that experimental science actually began in Denmark with the work of Tycho Brahe. Experimental science started when Brahe made his measurements of the movements of the stars and planets with great precision, with increasing precision, and this dovetailed with the theoretical work of Johannes Kepler at the same time in Europe. And it was really through their combined efforts that the enterprise in which we are engaged today found its beginning and we hope will never end. I thought I should just comment briefly about the title, which may seem presumptuous. It's actually partly facetious or self-mocking. The reason is that at least the older members of the audience will recall a similar title of a treatise written by a man named Francis Fukuyama. It was entitled The End of History, published about 30 years ago. As we all know, history hasn't ended. Nevertheless, I intend something serious by the title as well, which I hope to convey in the course of this lecture. I'm going to tell you a brief scientific story from my own experience, and then I will be so presumptuous as try and draw from it a larger meaning and connect it with something which I believe has much wider significance, and I hope indeed to many of you. Something which we might discuss further in the period that follows the lecture, which I much look forward to. So the scientific story that I'll tell you begins with the genome. It is, as you will know, most significant for its permanent, its conserved, its static nature. It is through precise replication of the genome without ever making a mistake that the information, genetic information, is transmitted from parent to offspring. This is the basis of heredity. The information in the genome, as has just been mentioned, is utilized through the process of transcription for the synthesis of RNA, and then translation by the ribosome for the generation of proteins, which are in turn responsible for all the form and function of a cell, indeed of every living cell. We used to think of this as essentially static in nature as well, that the structure of a cell was laid down, that its functional potential was established, and that it never changed. Remarkably, in the last couple of decades, it has emerged, the process is remarkably dynamic. Information from within the cell and also from outside is transduced through pathways leading to transcription, to regulate which genes are transcribed in what place at what time. The process is, as I say, remarkably dynamic. It reshapes the cell on a continual basis in response to environmental and internal information. It is a process which plays a central role in everything ranging from immunology and the response to invading organisms and disease, all the way to memory and function of the brain. The dynamic nature, the information processing takes place in the nucleus, which serves as a kind of central switchboard. The information processing, which leads to the control of transcription, is referred to as transcriptional or gene regulation. And that is what I will primarily speak to you about today. Now, gene regulation begins with the genome itself, and indeed, as has been mentioned, especially with the structure and organization of the genome. When I was about the same age as most of the young people in the audience, I proposed on the basis of X-ray and biochemical studies the wrapping of DNA around a set of 8 histone molecules and a particle that has come to be known as the nucleosome. The nucleosome serves as the fundamental or organizing principle, the basis for packaging all DNA in eukaryote genomes, but it serves an even larger purpose. Some years later, we discovered the nucleosome interferes with many DNA transactions, so the wrapping of DNA around histones in the nucleosome prevents the initiation of transcription, an observation we first made from studies of transcription in a cell-free system, so in vitro. But soon after, it was shown the nucleosome subserves a similar function for the control of transcription in vivo. The nucleosome then serves as a general gene repressor. It prevents expression of all the many thousands of genes in eukaryotes, except those whose transcription is brought about by specific positive regulatory mechanisms. Now, we and others have investigated those mechanisms, and much progress has been made over the past 30 years. I won't talk to you about that aspect today, but rather pass immediately from what happens when the nucleosome is removed, the DNA is unraveled and made available for interaction with the transcription machinery. And in our case, we focus especially on the RNA polymerase II transcription machinery for a reason that is well known, I'm sure, to many of you. RNA polymerase II is responsible for all synthesis of the messenger RNA that codes for proteins in eukaryotic cells. And as the first step in the pathway of gene expression, polymerase II transcription is a focal point of cellular regulation. Most of the signal transduction pathways that I alluded to in a previous slide culminate in the control of RNA polymerase II transcription. Indeed, it is the intricate regulation of polymerase II transcription that underlies cell differentiation and development. The machinery for transcription extends beyond the RNA polymerase itself. There are many additional components. We and others discovered what are called general transcription factors that go by the letter names B, D, E, F, and H. The polymerase alone is incapable of unwinding DNA and the synthesis of RNA, as I will show you in a snapshot at atomic resolution in a moment. But in the absence of general factors, the polymerase is unable to start the process. It can't find a gene. It can't begin transcription. The most complex of the general factors rivals in size and complexity the RNA polymerase, and it plays an essential role not only in transcription, but in 2 other seemingly unrelated but equally important cellular processes: DNA repair and control of the cell cycle. In respect to DNA repair, it may astonish you to know that the many possible insults to our DNA result in on the order of trillions or even thousands of trillions of DNA lesions in every individual every day. And the reason why these do not have profoundly deleterious effects is because of repair by enzymes that correct almost all such mutations. Principal among these is the enzyme general transcription factor H. Evidence for this comes from humans mutant in this factor, which is the cause of a disease, as I will explain in a moment. First, I must clarify there are 2 kinds of repair process, one called base excision repair, which involves recognition of damage, then excision of the damage to leave a gap in DNA, which is finally closed. The other more prominent form of repair, nucleotide excision repair, also involves recognition of damage, excision of a larger segment of DNA that includes the damaged region, and then finally, once more, filling the gap to restore the DNA to its original undamaged condition. Now, as I mentioned, there are mutations in humans that interfere with nucleotide excision repair. They are alterations of the general factor H and the disease that is a consequence called xeroderma pigmentosum. It results from extreme sensitivity to UV light that causes DNA damage, and that leads in turn to a high risk of skin cancer. And the only remedy at the moment is protection by the use of special UV opaque apparatus. Now, there are additional lesions that affect proteins, such as so-called CSA and B, that couple transcription to repair. They lead to a disease called Cockayne syndrome, also characterized by sensitivity to UV light, but in addition, which leads to neurological abnormalities and progeria, which is premature aging. Now, the general factors, as I have mentioned, interact with RNA polymerase to enable the initiation of transcription. Gene regulation is due to molecules that are either positive acting, so-called gene activator proteins, or negative acting, gene repressor proteins. These enter the nucleus of a cell. They contact recognition sites called enhancers in front of genes and impinge upon the transcription machinery to turn a gene on in the case of an activator, or to prevent its transcription in the case of a repressor protein. My colleagues at Stanford discovered some years ago there is an additional component required for communication between an activator protein and the general transcription machinery. Because it subserves an intermediary role, it goes by the name mediator. Especially my colleagues, Stefan Bjorklund, now a professor in Umeå, and Young-Jun Kim, presently in Korea, reported in 1994, mediator is made of dozens of proteins. It has a total mass greater than 1 million molecular weight. Over the years as it has emerged, mediator receives and processes all transcriptional regulatory information. Following information processing, delivers an appropriate signal to the RNA polymerase. And this occurs in the entire range of eukaryote organisms, from single-cell creatures like yeast and other fungi, to the most complex like humans and higher plants. Aberrations of regulation by mediator underlie a wide variety of human disease, including infectious disease, and, as this slide indicates, cancer. So here you see diagrammatically the 31 protein human mediator, and virtually every component is implicated in a variety of malignancies. It is interesting that in very many cases, for example, breast cancer, bladder cancer, the aberration of mediator is an overexpression, a superabundance of one of the components of the mediator complex, not a mutation of a component of the complex. And I can suggest, if someone would like in discussion later on, why this kind of aberration, simply a superabundance of a subunit of the mediator complex, should have such a high association with cancer. So in the case of both breast and prostate and bladder cancer, a particular subunit of mediator is overexpressed in more than 50% of all individuals suffering from the disease. To summarize then, the machinery for transcription, polymerase, the general factors, and mediator, constitute nearly 60 individual proteins that assemble in an object of greater than 3 million molecular weight, so larger than a ribosome particle, at every gene prior to every round of its transcription. Francis Crick, who you heard mentioned was one of my teachers, was supposed to have said, if you wish to understand functions, such as we do of mediator, study its structure. As I often say, when I made that remark to my father, he said, if you wish to understand structure, study function. Well, regardless, my colleagues and I have pursued the structure of the transcription machinery, and I'm going to tell you one of the findings which serves as a prelude for the connection to disease that I will explain later on. That one finding comes from investigation of the structure of the RNA polymerase itself. And this we did over a period of years by isolation of the enzyme in the formation of large single crystals. And these in turn could be solved by X-ray diffraction because the protein was at least 10 times larger than any previously studied. We required a source of X-rays much more intense than those previously utilized for this purpose. And they became available to us in the year 2000 from photon physicists at the Stanford Synchrotron radiation laboratory. The Stanford Synchrotron lies at the end of a 1-mile-long linear accelerator that you can see here in the distance in the background of the diagram. The electrons accelerated that 1 mile of length, then circulate around a ring, and in consequence of that motion, they emanate X-rays, and these are of especially high intensity. It took us 10 years to purify and learn to grow suitable crystals of the enzyme. It took another 10 years, 10,000 liters of cell culture from which the enzyme was derived, and one graduate student went into the solution of the problem. What you see here is my longest serving associate, Dave Bushnell, lowering a graduate student, Ken Westover, into the 100-liter fermenter to repair a valve at the bottom. Ken was a medical student and graduate student at Stanford, a so-called MSTP student. He went on to complete the requirements for both graduate and medical degrees, then did clinical study, so was a resident and a research fellow at one of the Harvard hospitals in Boston, and is now a clinician scientist at the University of Texas Southwestern in Dallas, Texas. He's a very fine scientist. Ken was at the time also a gymnast, and so slender and could fit into the fermenter to repair the valve at the bottom. And I often say, I wonder where we'll ever find another student of his caliber. So anyhow, we solved the structure of this enzyme by X-ray diffraction. We located the positions of almost 30,000 carbon, nitrogen, and oxygen atoms to near atomic resolution, arriving at the picture that is shown in this slide. Here, each of the individual subunits in a different color is displayed in the form of what is called a ribbon diagram that indicates the path of the polypeptide chain of the particular protein. The one that is colored purple at the lower left represents the 5th largest subunit of this giant complex. It is a 25,000 molecular weight protein, so a typical sized protein, which gives you a sense of the size and the complexity of the entire object. I'll rotate the picture around to a view down the central channel that I will return to in a moment. The first thing you wonder when you look at this enormous complex, the RNA polymerase structure, where does the gene DNA enter for transcription? From what point does the product messenger RNA molecule exit? Well, you might imagine that the gene DNA enters through this capacious central channel that I called your attention to a moment ago, that leads to the point marked by this pink sphere, which symbolizes the location of a magnesium ion required for catalysis, and so that identifies the active center. We proved that the DNA enters at this point, and RNA from the location of the magnesium ion by solving the structure not only of the enzyme alone, as you see here, but also the enzyme in the form of an actively transcribing complex with the gene DNA present and the RNA in the act of synthesis. I'll show you that in a moment, but first a color code to the slide that follows. It had been known for many years that about 15 base pairs of the DNA double helix are unwound in the form of a so-called transcription bubble at the active center of any polymerase engaged in a process like transcription. It was furthermore known that some 8 to 9 base pairs of RNA-DNA hybrid are centered within the bubble, with the growing end of the RNA adjacent to the catalytic magnesium ion, 3 residues from the single-strand, double-strand junction. In this slide and those to follow, the strand of the DNA that directs the synthesis of RNA is blue, the opposite strand is green, and the RNA is always red. The next slide is a movie. It's made of only 2 frames. Each frame is an actual crystal structure. And the one you see here, the first frame, the structure that I showed you already, that of the enzyme alone, viewed down the axis of the central channel, and with the protein almost all in one color instead of a different one for every subunit, with the exception of this part that is gold for a reason you'll understand in a second. The movie is produced by an alteration between 2 frames, this and the one of the structure of the transcribing complex. And what you appreciate from this movie, 2 things. First, the DNA in blue and green and the RNA in red do occupy the central channel. And second, this massive protein element colored gold swings over the DNA and RNA in the course of forming a transcribing complex. It serves the purpose of a clamp. It holds the DNA in place. And that is essential because transcription of some human genes takes many hours. And if the DNA is released, if the grip of the enzyme on the gene is lost at any point, the whole process is a failure. The purpose is defeated. Now what I'll do is just rotate the picture around. Sorry. I've removed some of the protein from the front to give you the best view of nucleic acids as they pass through the polymerase. The DNA here entering from the right. The structure reveals it as a double helix that unwinds 3 residues before the active center, after which there is a sharp bend in the template strand that codes for the RNA, in consequence of which this next base of the DNA is flipped, points down towards the floor of the active center for readout in transcription. That base is seen still paired to the ribonucleotide just added to the growing RNA chain. There are 8 more RNA-DNA hybrid base pairs that are revealed in the structure. You will have noticed that the axis of the entering DNA double helix is at nearly right angles to that of the exiting RNA-DNA hybrid helix, for a reason that I could explain if anyone is interested later on. But for now, what I'd like to do is remark on the significance of the structure and to tell you one important lesson that we learned from it. We have over the years learned many things from investigating this and related structures, but there's one point in particular that I wish to mention, and that leads to really what is the last message of my lecture. So what I'll do in the next slide is zoom in. I'll remove most of the protein, so you will see only this portion of the template strand, a bit of the RNA, and one marker, one element of the protein colored green. Here's that element colored green. This is the structure from the previous slide with the DNA and RNA, and it differs only in one respect, and that is that the nucleotide just added to the growing RNA chain, still paired to the template base in the DNA, is highlighted in yellow. Now, in another structure that was actually determined by Ken Westover, who I showed you repairing the fermenter in one of the previous slides, he could observe a state that results from movement of the DNA RNA across the enzyme surface, taking the nucleotide just added to the next position and exposing the addition site for entry of another nucleoside triphosphate. So that nucleotide has moved forward, another base is in position to direct synthesis of another step of the RNA transcription process. The nucleotide that enters binds in what we call an E or entry site and then rotates into the addition site for addition to the growing chain, bringing us back to where we started. The structure shows how the nucleotide is selected in part through pairing to the base in the DNA chain. But we know that the energy of that interaction is insufficient to account for the remarkable fidelity of transcription. The accuracy of this process is the essence of gene expression, of the correct readout of the genetic code, and it exceeds by at least a factor of 1,000 what could be accomplished through base pairing alone. We learned in years later the reason is because of an additional protein element. So this shows the one that was colored green in the previous slide. Here's that additional element that we call the trigger loop, and it can be seen to contact the entering nucleoside triphosphate before addition to the growing RNA chain. It contacts the base of the nucleoside triphosphate. It contacts the triphosphate portion itself, and indirectly, not shown here, the sugar moiety in between. It is the contact with the second of the 1, 2, 3 phosphates that is of particular significance. That contact by an amino acid of the trigger loop delivers a proton to the phosphate, which will depart following cleavage of the bond to the alpha, which in turn follows the interaction and chemical addition of the growing end of the RNA chain to that phosphate to complete RNA synthesis. So the delivery of that proton from the trigger loop quite literally instigates or triggers the process of RNA synthesis. It is the precise alignment of the nucleoside triphosphate that enables the proximity, appropriate proximity and orientation, and thus the delivery of the proton to trigger the process. That alignment is only suitable if the correct nucleoside triphosphate is present, and not if any of the other 3 possibilities obtains. This then is the basis for the fidelity of transcription. It is the most fundamental feature. Remarkably, as I will now explain, this most fundamental aspect can be exploited for a practical purpose. And that relates to what is a major unmet medical need, namely the treatment of viral disease. I show you here some examples of well-known viral diseases for which there is either no medication available or only a very inadequate medication. So the examples that I will mention are hepatitis virus, and in particular hepatitis virus C. I could also have mentioned hepatitis virus B. Flu, well known to all of you. Human rhinovirus, which is the cause of most common colds. And then perhaps less well known to many of you, as I will shortly explain, dengue virus and norovirus. So dengue virus is a problem of increasing significance. It used to be most common in the developing world, but with world travel has now become endemic also in North America and will doubtless soon be a concern in Europe. Norovirus is not only the most common cause, as the slide indicates, of diseases due to contaminated food. It's the reason why cruise liners often abort their voyages, because everyone on the ship becomes sick. A single particle of norovirus is sufficient to cause a serious infection, which can very often be fatal. Now, in the case of hepatitis virus C, there is medication on the market. There are pills that target a protease essential for viral multiplication. But these are really quite unsatisfactory. Not only do you need to take more than a dozen pills a day, but they cause very serious side effects. A further difficulty in treating not only HCV, but many viral diseases, there is not only one form of the virus, there are multiple forms. And beyond that, the virus evolves very rapidly. And almost any medication that can be created soon suffers from resistance evolved by the virus to the treatment. The basic research that I've told you is relevant to this problem because of the importance of the viral replication enzymes to the propagation of the viral disease. So the viral replication enzymes, and in particular the RNA polymerase of more than half of all the viruses that I've mentioned, may be viewed as the Achilles heel. And the reason is that in the simplest case, a virus consists of nothing more than a replication enzyme, the genome, and a package within which they are contained. A key feature of these enzymes, which attracted my attention, is their remarkable conservation. Strange as it may seem, almost all viral replication enzymes are the same. They are close to identical in structure in all human viruses that possess RNA genomes and replicate in this way, which accounts for more than half of all human viruses. And because of that extraordinary similarity, we may hope to profit from the information that we have gained about the fundamental mechanism of transcription to devise medications for disease, and we may even hope to create medicines that are effective against not one but many viruses. Now, in the case of hepatitis virus C, there was recently introduced on the market a molecule from the Gilead Corporation. At the time when the slide was made, it was in phase 3, but it has now completed phase 3 and is an approved drug. It is, however, a nucleoside analog. It is a close relative of the substrate for RNA synthesis, of the nucleoside triphosphates that I mentioned. There have been many such drug candidates created in the past and entered into clinical trials, and with this exception, they have all failed due to toxicity for the obvious reason that nucleotides are used by practically every process in the body, and there is unavoidably a very narrow window of therapeutic efficacy between the amount needed for treatment and the amount that is harmful to the individual. The Gilead drug holds promise, but it's only been tested in some thousands of patients in a phase 3 trial, and it would be surprising if it did not suffer from some similar, from this kind of limitation as it is introduced into many more patients in the future. Now, the information that I have told you about suggests an approach for the creation of a non-nucleoside inhibitor of an RNA polymerase that could be especially effective for the treatment of viral disease. Where better to look for a source of a non-nucleoside inhibitor than in nature? Indeed, the best possible approach to the treatment of such a disease has been devised by nature itself. And this slide shows a result from our own work so it shows a crystal structure of the well-known mushroom toxin, alpha-amanitin, bound to the trigger loop of RNA polymerase that I described only moments ago. Alpha-amanitin is produced by the death cap mushroom. That, as many of you know, is the reason why only a well-trained person should ever dare to eat a mushroom out of the forest. If you make a mistake and you eat a death cap mushroom, the only cure is liver transplantation. There is no resistance. Nature has discovered a way of creating a poison against which resistance cannot arise. And it is done so by targeting that key catalytic residue of the trigger loop. So alpha-amanitin binds that residue and in that way inhibits RNA polymerase. There is no escape from such inhibition. No resistance can arise because if the residue is changed, well, the inhibitor won't work, but the enzyme is dead. So the purpose of the inhibitor or the escape from inhibition is defeated. In recent months, we have devised a series of small molecules that accomplish the same purpose with respect to viral replicative enzymes. So here's a list of some of those molecules. And you see that the very first ones that we have made are highly inhibitory to a viral replicative enzyme, and they are pan-viral in their activity. They're equally effective against all the viruses that we've tested: hepatitis C, flu, dengue virus, the common cold virus, human rhinovirus. So this holds out the spectrum of creating antiviral drugs that not only show a very high barrier to resistance, but will also be broad spectrum, like the antibiotics that are widely used for the treatment and that have largely eradicated bacterial disease. Now, in the last few minutes, I will do what I promised at the outset, and that is be so bold as to extrapolate from this limited experience to something far broader. And not meaning to be immodest, but to place it in a broader context of what I would refer to as really the history or the evolution of modern medicine. Perhaps not all of you, perhaps even only a few of you would be aware that modern medicine, as we know it, the medicine we take for granted, is only about 100 years old. A little over 100 years ago, all human disease was attributed to an imbalance of humors, and the only treatments were bleeding or violent purgatives. It was only with discoveries made by Pasteur and others in the latter part of the 19th century, and then the emergence of medical science at the beginning of the 20th century, the situation was transformed and medicine developed into what we know today. If I were to ask any of you what were the most notable advances responsible for the development of modern medicine, I'm sure somebody would mention, one of you would think of X-rays for both the diagnosis and also the treatment of disease. I'm sure somebody would suggest what I have just alluded to, antibiotics, which, as I said a moment ago, have eliminated or largely eradicated bacterial disease. Someone might think of non-invasive imaging, MRI, for example. Genetic engineering, which is really the basis for a large fraction of the new drugs that are now under consideration for approval by the Food and Drug Administration in America. Now, if we just look at a couple of these examples, X-rays and antibiotics, in a bit more detail at a moment, consider where they came from. X-rays were discovered by Wilhelm Röntgen around 1898, 1899. Röntgen was a physicist, an interesting man actually. He was thrown out of school for drawing caricatures of his teachers and never allowed back. He nevertheless rose to be a leading professor of physics in Germany. Röntgen was interested in the discharge of a cathode ray tube. And in order to see it better, he darkened the laboratory. And then he noticed, when the discharge took place, a faint glow on a fluorescent screen elsewhere in the near vicinity in his laboratory. And he was curious about what signal could pass from the cathode ray tube to the screen, so he put his hand in front and he saw the image of his skeleton. He saw the bones of his fingers. Within 2 years, the discovery of X-rays was widely employed in medical practice. And in 1901, Röntgen won the first Nobel Prize in Physics. Now, similarly briefly, just a comment about penicillin. Almost everyone knows the story of the serendipitous discovery by Alexander Fleming of a mold on a petri dish that was otherwise covered with bacteria, but a halo around the mold where bacteria died. And this is supposed to have been the origin of penicillin in medicine, but that's not true. Fleming did make that observation and he published it, but he could never progress beyond that observation. And his publication went unnoticed or was largely forgotten. And it was only decades later that Florian Chain, interested in the synthesis of bacterial cell walls and wanting to discover the mechanism, took advantage of the work of Florey before them. Chain, who was both a scientific and a musical genius with a photographic memory, could recall everything that he had seen in the literature, including the paper of Fleming, and brought it to bear on their research, showed that the element responsible for the killing of bacteria found by Fleming interfered with the synthesis of a bacterial cell wall, and it was finally through the work of Florian Chain that penicillin came into use for therapeutic purposes. So my point is that these major advances, responsible for much of what we know and I should say largely take for granted as the basis of medicine today, were due not to scientists trying to solve a medical problem, but rather to the pursuit of knowledge for its own sake. So the moral of the story, the lesson from this history, is if you want to solve a difficult problem, practical problem, don't try and do it directly. The lesson is counterintuitive. Instead, pursue a curiosity about nature, and the solution of practically every problem will eventually be found. The solution for viral disease, I suggest, and I am confident eventually cures for cancer, and eventually even a cure for aging. And with that, I thank you for your attention. Thank you. Thank you.
Thank you very much for a very inspiring lecture. We now have a question and answer session, and it's not just a few questions. It's we have about half an hour, so I will suggest, Roger, that you take a seat.
Pleasure.
And you in the audience should prepare yourself for the questions you would like to ask Roger. It can be to the science, it can also be to how it is to be a scientist and how to be a Nobel Laureate. So please think about it, and please take a seat.
Thank you.
So, anyone volunteering for the first question?
Thank you. I'm Kåre Nielsen, Aalborg University. I was just wondering, since you showed the table of your leading compounds, you can say you showed the efficacy of IC50 values for the viral polymerases. But of course, as you also mentioned, the therapeutic window, do you have any, of course, I assume you have ideas of what is the inhibitory efficiency of the eukaryotic polymerases, or most notably the human polymerase as well?
So these are molecules which are only effective against the viral enzyme because they react with an amino acid of the viral enzyme not present in the human one. And they're actually composed of only two small parts, both of which are non-toxic to humans. One part is actually found in cruciform vegetables. It's abundant in cauliflower and broccoli. So there's a good chance that the pursuit of these first molecules that we've made will lead to something useful.
Hi, I'm Casper from Copenhagen University as well. I was wondering, you stated that alpha-amanitin no resistance is possible to alpha-amanitin, the polymerase II inhibitor. Is that really true? I mean, the fungi in nature, are they not eaten by, let's say, other fungi or snails, for instance? I mean...
So any eukaryote organism that consumes a death cap mushroom is susceptible to the fatal effect of alpha-amanitin. We don't actually know why the death cap mushroom itself survives in the face of the abundant alpha-amanitin. It is capable both of synthesizing and accumulates in its tissues. There is the possibility of a resistant mutant of RNA polymerase, and that is because the contact of alpha-amanitin is, although primarily through the amino acid that I showed, also to other features of the enzyme surface, and those can be changed and create resistance. But I showed that example for purposes of illustration that served as a guide in our own creation of a molecule that makes no such additional contacts, but only with essential features of the enzyme, and against which we hope, in this particular case, truly no resistance will arise.
My name is Susanne Mandrup from University of Southern Denmark. I wonder if you could say something about the complexity of the mediator and the advantage of having such a high complexity, the differential necessity for the individual subunits, etc.
So it is remarkable how many components are found in the mediator of virtually every organism. In human cells or in human beings, the composition varies slightly from tissue to tissue. There is a core set of some 20 or so, but then many of the additional components are actually tissue specific. Likely the reason is because there are thousands of gene activator and repressor molecules. These are small proteins consisting only of a part to bind the signal sequence in the DNA, and then a part to interact with mediator. In order for a mediator to make a productive contact with thousands of molecules, it needs itself many components to provide extensive surface for such interactions. Not only interactions with different activators for different genes, but even multiple activators and repressors for a single gene. So a typical human gene will have associated with it as many as dozens of regulatory sites which bind as many activator and repressor molecules simultaneously to control the time, the place, the extent, and what have you of transcription required of that gene and that tissue in the organism. So mediator in some way can make multiple contacts, very many contacts, in that way receive information, then process the information as a kind of molecular computer, and in some way arrive at a unique signal that it delivers to the RNA polymerase to control transcription.
Yes. Michael?
Michael Overgaard from Aalborg University. Thank you for an inspiring lecture. I just wanted to hear if you would comment on the fact that you list aging as a disease.
Aging is, as it were, a condition. And many of us believe that it is genetically programmed. There is some evidence for that. Even ordinary observation would lead one to believe it is the case. We have almost the same genome as a mouse, but a very different lifespan. We don't know the reason why, but it would seem to make obvious sense in terms of how long we need to live for the benefit of survival and evolution of the organism and the species. So if you accept that it is very likely genetically programmed, then it is also very likely we can intervene and manipulate or alter, in some way control that program. And in that sense, I refer to it as a disease or a condition that we hope to modulate or ameliorate in some way.
Hi, my name is Peter Brothers from Copenhagen University. I was wondering about the clamp you were showing that has to swing onto the DNA to hold it during the process of transcription elongation. Is it known whether at transcription termination that clamp must swing out? And if so, what are the mechanisms that are responsible for that?
So what you ask is actually a question that concerned us a lot. From the time in our studies that role of the clamp first emerged, so when we first became aware of it from the crystal structure that I showed. And you would think that the initial interaction with the gene DNA would require swinging out of the clamp, and then it would only come to cover the DNA in a subsequent step. We still don't know for sure. We're still trying to find out. And it's possible that such motion is not required, that the DNA can even be inserted without opening. We just don't know.
While we're getting the microphone around, maybe I could ask you, is there any resemblance between the RNA polymerase and the DNA polymerase, and would they have a common evolutionary origin?
So it's interesting that, first of all, I referred to the conservation of the viral RNA polymerases. They have nearly the same structure as viral DNA polymerases. So I said about half of all viruses use an RNA polymerase for their replication. They have an RNA genome. There are many viruses that have a DNA genome, but they use a DNA polymerase that's also nearly identical in structure and well conserved in evolution with the RNA polymerases. And so a similar approach to what I mentioned could be employed in that case. This entire family of enzymes differs fundamentally from the molecule whose structure I showed and that we have studied all these years. So the viral polymerases are made of single protein, for the most part a single protein subunit with a characteristic structure. The molecule I reported on, the one I showed you in these slides, belongs to a different family. It was separately evolved. As you will recall, it's made of a dozen protein subunits. The structure is altogether different, but the mechanism is the same. And therefore, that conserved feature of the mechanism, which in the case of the enzyme we study is a histidine amino acid side chain, performs a role also needed by the viral polymerases, except with a different amino acid. It is because of that difference that we can make a drug which is selective and only effective on the virus and not on the human. There are then two distinct families of nucleic acid polymerizing enzymes. Those represented by the viral polymerases, and both DNA and RNA, and that represented by the eukaryotic, in fact, bacterial and eukaryotic multi-subunit RNA polymerases. There is an additional family of enzymes, which are the DNA polymerases that are responsible for replication of the human genome and those of other cells. Again, very different in structure and independently evolved.
Thank you very much. There was a question down here. And okay.
Hi, this is Prash. I've just got two different questions. One is, what would be the fate of these polymerases in case of some mismatched repair system, especially in mitochondria? We seldom find MMR system in mitochondria. That is number one. And number two, are there any specific cases, or to what extent does these polymerases or DNA, RNA interactions have got a role to play with proteins, protein-specific residues, or proteins having some amino acid residues towards the C-terminal? For instance, if there are proline-rich residues at the C-terminus, there is perchance of emitting more signal. There are some studies that are literally known where polymerases might really bind to some C-terminal proline-rich residues of a protein, thus emitting more signals.
So, in response to your first question about mitochondria, there is a molecule in humans similar to viral polymerases, and that is the main source of concern for specificity of an antiviral drug directed against a polymerase. That molecule is the mitochondrial enzyme. Both mitochondrial RNA and DNA polymerases belong to that family, which I referred to, mostly populated by viral RNA polymerases. However, the viral enzymes again use a different amino acid residue for the purpose of triggering the reaction. And so we can expect that what we have created that is specific for a viral enzyme will not attack the mitochondrial enzyme, and that's in fact been shown. So one will very likely avoid toxicity due to an effect on mitochondria. At the same time, it may interest you to know that the nucleoside analogs that have been employed as drugs, and even the one which is now finally on the market, are of grave concern with respect to mitochondria. So the toxicity in that case may not be immediately apparent. The nucleoside analogs diminish the activity of the mitochondrial enzyme, and this can have a consequence not in the first but in the second generation due to the necessity for multiplication of mitochondria and the creation of the oocyte, which is laid down in the ovary of the developing fetus. This is a worry about the drugs that have been used to treat HIV that people are not much aware of due to the extraordinary emergency with the rapid spread of the disease. These drugs have come into use and they are widely employed today. They do alleviate the condition. But people are not much aware of what may be a grave danger down the road when people who were exposed in the second generation beyond enter reproductive life. With respect to your second question, the only comment I can make that is relevant, the RNA polymerase whose structure I showed interacts directly with mediator for the control of transcription through a C-terminal domain that is rich in proline residues. And that interaction is unique to this RNA polymerase, and it is one of the key elements in the process of gene regulation.
Well, there's a question up here first, and then there's another one over here. So,
okay, yeah, thank you for an inspiring lecture. My name is Morten Sommer, I'm from DTU. You discussed some of the sort of serendipitous discoveries of science that had a big impact on medicine, and say that we should pursue sort of knowledge for knowledge's sake in order to make these discoveries. So there are also a lot of research for knowledge's sake that doesn't lead to sort of groundbreaking medical discoveries. Can you kind of qualify or give some insight as to which knowledge may have a higher potential to pursue in order to make some groundbreaking medical discoveries?
So I heard a scientist who I much admire named Georges Alferov, Russia's only living Nobel Laureate, a great physicist, now about 85 years old, say one time about, or quote, one of his own mentors, also an extraordinary physicist from the previous generation in Russia, who said, all science is applied, it's only a matter of time. And I believe that's true. I don't think there is any basic knowledge that we acquire that will not prove ultimately useful. It is only a matter of time. There are remarkable situations, such as the discovery made by Röntgen, that see medical application almost immediately. There are other discoveries, like those concerning are the molecular basis for nucleic acid synthesis, which were made in the 1950s, but it wasn't until the 1980s that they became the basis for genetic engineering. And in some cases, it may take even longer. The reason why I think it's so likely that all knowledge of human biology will ultimately prove useful is because every component of the organism is there for a reason, essential for some purpose. And we need to know about the details of its origin, its form, and its function in order to remedy a condition that affects virtually every feature.
Thank you very much. There's a question over here.
Yeah, hi, Liz, also from DTU. I was wondering, in your relationship between structure and function, as you study structure and recognize similarities and mechanisms, how do you structure your own thoughts, or maybe your research, in trying to generalize the findings from specific structures or repeated mechanisms to kind of allow you to increase in scale?
So there are a variety of ways that you can exploit such information. I'll just give you one example. For the development of antiviral drugs, we can, as I have mentioned, based on knowledge of the mechanism, create a molecule that is a starting point. It is effective, but it needs to be improved. In order to do that, we solve a co-crystal structure of that molecule with the enzyme. We are able readily to grow crystals of some viral enzymes, but not all of them. Because of the extraordinary conservation, there are a limited number of differences between any one viral enzyme and most others. We can take advantage of the ability to crystallize one, but see the structure of another by changing within the relevant region, just the active center, only those residues that differ between the molecule we can crystallize and the one whose structure we wish to observe. We create a kind of chimeric molecule that has a surface, has a shell that crystallizes readily, which we already know how to do, and a core which represents a different molecule, which is the one whose structure we aim to study in respect to a drug candidate or what have you.
And Susanne?
Thank you for a very inspiring lecture. I was wondering if you could give me a reflection on the treatment of cancer today. Where do you see we are? And also a little bit about the future. How do you think we will actually develop the treatment of cancer during the next years?
So I think first of all, the treatment of cancer today is accomplished either with leaving aside common chemotherapy, which of course involves treatment with poisons to almost kill the patient and one hopes completely kill the cancer, and also leaving aside the use of radiation, surgery, and what have you. There are two kinds of pharmaceutical approaches that are employed: small molecule drugs like Gleevec, and antibody drugs like rituximab and so very many others that you're aware of, Humira and so forth. There's a particular form of antibody therapy that has attracted a lot of attention in recent months. And that is the use of molecules which modulate the immune response and help to harness our own immune capability to destroy cancer cells and eliminate cancer tissue. So what are called anti-CTL1 antibodies, anti-PD4 antibodies attract a lot of attention and hold great promise. These are effective in some cases. So the first clinical trials of the anti-CTL1 antibody were done on metastatic melanoma, and the cure rate has been very impressive, close to 50% of all patients treated. But still 50% are in some way or other beyond cure by this or any other means, and so additional therapeutic measures are required. I believe that there's still an important role of small molecules. There are many targets for chemotherapy, for small molecule therapy, that have not yet been exploited, and many more will be discovered as understanding of the basic processes underlying the oncogenic state emerge from additional basic studies. The kind of approach that I mentioned for the treatment of viral disease will become possible as we understand better the mechanisms of the molecules involved in oncogenesis. But a notable example is RAS, which is, as you know, mutant in a very large fraction of all human cancers. And very many pharmaceutical companies and scientists engaged in the study of human cancer have sought small molecules effective against oncogenic RAS. Ken Westover, whose picture I showed you on that slide, who's now at UT Southwestern, has created a molecule by a very similar principle to what I said for our antiviral drugs, which is remarkably effective against one form of oncogenic RAS. And his work has just been accepted for publication in the Proceedings of the National Academy. You can read about it there.
I think there's a question over here. Yes.
Christian Pietras from Lund University. Your final sentiment about curiosity-driven research echoes those of Randy Sheckman at last year's Nobel Prize banquet. Do you feel that basic science is at risk from all the applied and strategic research initiatives being rolled out by funding agencies currently?
In America, where almost all the support for fundamental studies comes from the National Institutes of Health, the NIH, there's no question that significant funds have been diverted from fundamental studies to what is called translational research or even drug development. Within the NIH, something very foolish has been done, and $1 billion has been set aside for drug development within the NIH. No one in the pharmaceutical industry can understand why anything so foolish has been undertaken. The pharmaceutical industry is very capable of developing drugs, and they're doing it very effectively, AstraZeneca amongst others, all the time. The NIH doesn't know how to do it and never will. The total budget for basic research, such as I described at NIH, is only $2 billion a year, and now $1 billion is being devoted to this mad pursuit of something which NIH will never do as well as the pharmaceutical industry. So in that respect, yes, basic science is under threat, and we can only hope for some epiphany or change in direction or what have you on the part of those who lead the NIH.
Thank you for the very clear lecture. I'm Chris Workman from DTU. I'll take the bait on the question about overexpression of mediators. Can you say something about that?
Go ahead.
And one other quick question, because we're running out of time. Pol3, can you say a little something about why the genome is organized in a way that tRNA genes have to be present in many copies, whereas Pol2 targets tend to be present in 1 or 2 copies in the genome? So was there something fundamentally different about the way in which Pol2 and Pol3 are regulated?
So in answer to your first question, for which I thank you, it's easy to imagine why overexpression of a protein component of the mediator could result in a profound aberration of gene expression and, in one or other circumstance, an oncogenic fate. As I mentioned in answer to a previous question, we know that gene regulatory molecules, both activators and repressors, directly contact mediator. If one subunit of the mediator with which such contact is made is overexpressed, then there will be not only that same subunit associated with the mediator complex, but a superabundance of that subunit in a free form. The activator molecule will then mostly interact with that superabundance of the molecule in a free form and be diverted from contact with mediator. And in that way, overexpression of the subunit interferes with the normal pathway of expression. Again, either preventing activation or repression of the gene in question that is the target of that activator or repressor. The number of subunits of the mediator is relatively limited, so many activators and many repressors must use the same contact sites. And so it's not at all surprising that overexpression of one or more of the mediator subunits then will affect expression of a cancer gene. That hasn't been shown directly, but I think it's a reasonable hypothesis based on what we know. In answer to your second question about the other polymerases, so Paul II, devoted to the synthesis of, mostly to the synthesis of messenger RNA, has special characteristics required for the control of protein coding genes, the interaction with mediator, and all the other features that enable the extraordinarily intricate regulation. Genes that must be on in some tissues, off in another, or modulated in their level of expression according to the input of regulatory information. At the other extreme, one has, from the messenger RNA, one has the ribosomal and tRNAs. So all the machinery, all the RNA molecules that are involved in protein synthesis. And these are, of course, required in a superabundance, far beyond the level of any messenger RNA, in every cell, in every state, all the time. And nature has seen fit to create a streamlined process for the creation of those abundant stable RNA molecules, unlike the relatively rare unstable messenger RNA molecules. And so, in effect, what nature has done is to create an alternative form of RNA polymerase II, from which it has stripped the regulatory capability and in every way, and also removed the requirement for most of the general transcription factors. A simplified version that can enable the far greater production of the abundant tRNA and ribosomal RNAs for protein synthesis. For example, there's no factor H for Pol I and Pol III. They achieve the same purpose in a different way.
Very well. Henrik?
My name is Henrik Wigner. I'm from DTU. As we're approaching the end of the Q&A session, maybe a not entirely serious, but then again, question. When we find a cure for H, when should we start treating the patients? Assuming that there is a relation between wisdom and age, and maybe only in the light of the advancement of science. In your experience, when are we at the pinnacle of our wisdom when it comes to advancing science?
I will duck the question by saying what you, of course, know, and I don't mean to be patronizing in the response, that scientists have the task or the privilege of discovering information, but it's always the responsibility of society, including scientists, but only as equal members, to discuss and arrive at such weighty decisions. Won't really be a problem for me. It's not going to happen in my lifetime. I won't take part in that decision, so I can be forgiven for ducking your question.
Thank you very much. I think we are approaching the end of the session, and I would like to thank you very much for very inspiring answers, and you have definitely brought us along the road towards ending the diseases and the premature death of AIDS. Thank you very much, and let's give...
Thank you. Thank you. Thank you.